stress and there's a bad side of stress. And our goal is to try to understand, how does the brain interpret stress? What are the types of imprints that stress leaves on the brain? And how do these imprints, whether they occur at the level of a cell, a synapse, a molecule, a circuit, how do those imprints change future behaviors and future physiology? So we can go back almost 100 years, 90 years ago, when this gentleman, whose name is Hans Selye, who's a Hungarian who was at McGill University at the time, first defined and used the term stress. And so he said stress was this syndrome that was produced by a diverse, innocuous agent, right? Something that is not very appealing. And he said that all living organisms respond to stress. And he borrowed their term from engineering and from building, about the stress that's placed, how a physical weight places a stress on a piece of metal when it's bent, okay? So he said all organisms respond to stress. And in this respect, he said the basic reaction pattern is always the same, regardless of the agent used to produce the stress. So the concept that he put forward was that you must respond to stress, and the way you do it, regardless of the organisms, is kind of pretty much the same. And there is some truth to that, but there's also a lot of refinement that's happened over the last 90 years. So very broadly, what Selye was referring to was that stress triggers a change in our internal state, our physiology. So when you encounter a stressful stimulus, your heart rate, your blood pressure, your respiratory rate immediately increase, right? There's an increase in the release of specific hormones in your body, which we'll talk about later. But there's also a shift in how you relate with the world. There's also a change in your behavior. And all of those things happen in a very coordinated fashion. And one of the goals of our lab is to understand how this coordination happens and what the consequences are. But if I were to ask you if you're stressed, you would probably say, I am stressed, or you would say, I feel stressed. So when you say, I feel stressed, it's like saying, I feel angry, or I feel happy, or I feel afraid, or I feel sad, or I feel disgust, right? So stress, yes, is this response to challenge, but stress is also something else. We kind of view this almost like an emotional state. We don't talk about it that way, but I think we should talk about it as almost an emotional state. So it's this internal state that guides behavior, cognition, and mood. But is it an emotional state? We actually don't know. But what I'm intrigued by is that if it is an emotional state, we can use stress and models of stress to better understand the neural correlates of emotion. It's something that we still, as an entire field in neuroscience, don't really understand. We can't even agree on what an emotion actually is, let alone how the brain encodes specific emotions. So when we think about stress, we think about the brain. This is a section taken through the side, a sagittal section of a mouse brain. For those of you who have not seen a mouse brain in a picture before. And so this is just to say that there are many, many brain areas that are activated in response to stress, right? But they're not uniformly activated in response to any stress. So there's a generalizability, lots of the brain is activated, right? But there's also a specificity. This is an example of what that specificity might look like. If there's a physiological stress, let's say you're dehydrated, right? Or you're really deprived of food, you're starving. There's a set of pathways in the brain that are activated in response to stresses like that. If it's a psychological stress, right? There's a danger, a threat, something is going to have a negative impact on you right away. That psychological stress is interpreted by different brain circuits, right, than the ones that interpret physiological stress. But what I want you to pay attention to is they all kind of converge on a tiny little area of the brain that sits just at the base of the brain, in us, just above our palate, right? Just at the bottom of our skull. And that area is called the hypothalamus. And specifically, there's one population of neurons, about 3,000 neurons in the mouse, probably 5,000 to 8,000 in humans, not that many considering we have over 1 billion neurons, that seem to be responsible for coordinating the response to stress. It's a lot of responsibility for not that many cells in your brain. So this is how these cells work, and this is what the textbook will show you. These cells, which are known as CRH PVN neurons, CRH for corticotropin releasing hormone, that's the hormone they release. PVN, because they are located in the paraventricular nucleus of the hypothalamus. So these CRH PVN neurons make this peptide called CRH, and they project down to the stalk of the pituitary, which is a little stem that hangs down from your brain, connecting your brain to the pituitary, which is a gland that releases hormone. And these cells release their hormone into the blood vessels of the stalk, where it's carried down to the anterior pituitary to trigger the release of a second hormone called adrenocorticotropin releasing hormone, ACTH, which circulates in the general circulation to act on the adrenal gland to release glucocorticoids. Cortisol in us, corticosterone in mice and rats. And so over the last 60, 70 years of stress literature, we've come to learn that stress triggers an increase in the release of CRH, which in turn triggers an increase in the release of ACTH, which causes an increase in circulating cortisol or corticosteroids, right? And this time frame from the stress being time 0, even if it's a brief stress, this time frame can be like 90 minutes, okay? So there's a very rapid signal, a rapid processing, but then this very slow onset of this hormone release and accumulation. This is this hormone building up in the brain and acting on the brain. Even though it takes a long time, the field has largely believed that glucocorticoids are responsible, or these corticosteroids are responsible for the effects of stress on behavior. So the classical loop is that stress triggers activation of this system, and these corticosteroids then feed back, act on the brain, and modify behavior. There is some truth to that, but there are also some rapid behaviors that we engage in, mice engage in response to stress, that have nothing to do with this hormone and speaks to the different roles that these cells in the hypothalamus play. So there's the classic axis I just talked about. Over the last kind of decade or so, our lab and many, many other labs have shown that these cells do a lot of processing of different types of stress information. They change how they respond to subsequent stressors. They change how their synaptic inputs, so the connections from other cells to these cells, can be modified. They change how they remember different types of information, right? So we're not going to talk very much about that. That's just to say that there's a lot of processing that happens way deep down in the brain that has nothing to do with the cortex or the hippocampus or other brain areas. And over the past decade or so, we've also started to figure out that these cells themselves coordinate a lot of other stress-related behaviors. So we showed 10 years ago that they're very important or essential for anxiety-like behaviors and for grooming, which is a type of stress coping behavior in mice. Over the last few years, we've shown that these cells are really, really important for this phenomenon known as social transmission of stress, where I can transmit stress to you and you can transmit stress to somebody else. We're not going to talk very much about that, but it's just a super cool observation. And then the other thing that we are going to talk about today is the role of these cells in avoiding threat, right? Engaging in specific behaviors that allow, in this case, mice to avoid threat. So this talk is going to be divided into 3 very different parts. Well, 2 are linked, the third is not. But they all have these CRH neurons as the kind of stars of the show. And we're going to manipulate these cells. We're going to look at what they do. And we're going to talk about how they engage in different types of behaviors. The first part of the talk we call Should I Stay or Should I Go? It's basically about how these cells regulate escape type behavior in response to threat. The second part of the talk that follows on is learning on the fly and talks about what we think is a new form of a trial-dependent learning in which these mice show very rapid learning in response to their interactions with stimuli. And then the third part of the talk we call it stranger danger, and that is how mice and these cells respond when we introduce them to other mice they don't know. These are the tools of the trade. We use a lot of different tools, and sometimes I just kind of fly through them, but I think it's important, and we'll come back to them. So we use approaches that allow us to make these cells glow with non-native proteins, and whenever the cells increase their activity, we can get a signal from these cells. And we can get two types of signals. One is a signal from the entire population of cells, and we use a technique called fiber photometry to do that. Right? So fiber photometry collects light from these cells that's emitted when these cells become activated, and we can see that signal. We can also use something called miniscopes, which is a tiny microscope that weighs about 2 grams that is on top of a mouse's head. And while the mouse is doing whatever the mouse wants to do, we can record from single cells, and not just any single cells, but specifically CRH cells deep in the mouse brain. We use an approach called chemogenetics, which allows us to target these cells with a specific receptor, which when activated silences these cells. And then we can use a tool called optogenetics. And what optogenetics does is it allows us to express a protein in these cells which is responsive to light, okay? And the specific protein that we express that we'll talk about in this talk turns these cells off. So when you turn on a laser with yellow light that you put into the brain, these cells switch off. So these are the cells being active, they're spiking, you turn on yellow light, the cells shut off. So you can basically use light as a switch to just turn cells on, turn cells off, okay? So we'll come back to these, but I just wanted to put them out here so everybody understands. Okay, so this work was done primarily by Nuria, and it's based on, I'm sure many of you have seen this iguana snake video, right? So the iguana hatches, everyone is nodding, this is an awesome video, actually. The Snoop Dogg narration one is also great. So the iguana hatches, and immediately snakes start to attack it, and it runs from the snake. The iguana has never in its life seen a snake, right? How does it know that it should run? Why does it not just hang out and go, hey snakes, like what are you doing today? No, it doesn't. Immediately it starts to run and tries to make an escape. And although I have no horse in the game, you're really supporting the iguana to get away in this. It's really amazing. Anyway, this is just to say that most organisms have innate programs, right, that allow them to escape threat. I'm going to talk about one of these in mice. In mice that appear to evade what is a looming, advancing predator from the sky, like a hawk or a falcon, even though for hundreds of generations, these mice, which are bred in a lab, have never, ever been exposed, right, to this predator. We started this experiment because we made a very, very simple observation. When we first started doing fiber photometry, Tomasz made this observation, that as soon as he got near the mouse with his hand, these cells would turn on. And then as soon as he was holding the mouse, put it down, these cells turned off, right? And so we're like, that's weird. We're not interested in hand-holding of mice. And so what we realized is what the mice were responding to was this evolutionary program, right? Where they would respond to threat that comes down from above. So Nico Tinbergen is a behavioral ethologist. Actually, I was talking about him earlier today. And I love his work on behavior. And this is one of my favorite quotes in science that says, contempt for simple observation is a lethal trait in any science. So not wanting to anger Nico Tinbergen, we thought, let's just follow the simple observation and let's see where it takes us. So let me just set up this story a little bit to put it into our context, okay? So let's say you're walking down the street. It's dark. It's like broken windows. It's an alley. You're kind of in this unknown area. And you start to hear footsteps, right? You have to think about what you're hearing, and then you have to make a decision, and then you have to take some type of action, okay? So you detect a threat. There's footsteps that are approaching, right? Then at some point you have to decide, what am I going to do? And then you have to decide, am I going to run? When I'm ready, I'll run, okay? Then you execute, right? So you made your decision, then you execute your action plan. You start to run. And then you run until what? You don't hear footsteps. You turn around and look. No, I would argue you run until you feel safe, right? That might be you find a door and you run inside. You might turn a corner. There's a store that's open. You see your friend. So you do all this, right? So you engage, you listen, you make a decision, you take action, and then when you feel safe, you stop running. So we think there must be somewhere in your brain something that signals danger and safety, that tells you now's the time to go, now's the time to stop, okay? And that's what we're gonna explore a little bit. But we're gonna explore it in this mode. This is actually a real photo. This is not ChatGPT photo. This is an owl descending on a helpless, I think helpless mouse. Maybe the mouse gets away, I don't know. And this is kind of our model. In a lab, it's not as cool. It looks something like this. It's an arena in which there's a shelter that the mouse has had some time to become familiar with. And over top of it, we have a screen. We just turn a screen upside down. And there's a shadow that shows up on the screen. And then the shadow expands, okay? And expands. So that it shows up for 3 seconds. It expands over 2 seconds, and then it's just steady for 3 seconds, right? It's an 8-second protocol. Okay, so it looks something like that. We call this the danger zone. When mice are in this area is when we turn the shadow on. We want them far away from the shelter. They come to that zone, we turn on the shadow, we watch what they do, okay? And so the mice have to make this exact same decision that I just talked about with you in a dark alley when somebody's chasing you. They detect the threat, right? They have to make a decision on when am I going to run, and if so, when? And then they execute, right? And they execute that plan until they feel safe. So mice show 3 different responses in our hands. They either escape, or they freeze, which is also a defensive behavior, which is largely linked to not wanting to be detected, right? Or they show no response, in which case there's a shadow that happens and the mice are just like, whatever, and they don't do anything. So this is what happens in our first set of trials that we did. These are 5 trials, 1, 2, 3, 4, 5. That's a mouse, and these are all the different mice. And you can see, without overthinking this at all, what you mostly see is blue, right? Blue is escape, blue will always be escape, green is always freeze, and gray is always no response. So the majority of trials, you see escape, okay? So about 80% actually of the mice show escape responses, right? This just plots the trajectories of where the mice were. They can be all over the place. Most of them still show an escape response. What about these CRH neurons? What are they doing when the mice are escaping? So when you show the shadow, each of these lines is a single cell, and the colors is a heat map. So the brighter the color, warmer the color, the more the cell is active. So red means the cell is really active, right? Purple means the cell is very quiet. And you can see that as you present the shadow, many of these cells become really active, right? If you add all this information up as numbers, you get a signal that looks something like this from all of these mice, right? There's an increase in the activity of these cells, which then drops. What's interesting is that this increase in activity goes up, the mouse reaches the shelter, and it comes down. So it looks like they're responding to potential risk, and then when the mouse feels safe, they're switching off. What's also interesting is that they only turn on when the mouse escapes. So they seem to turn on about a second or 2 before the mouse escapes, and then once the mouse has reached safety, they turn off. And so I'm showing you all the trials in which there was an escape. You can see there's a really big increase in the activity. And these are the trials where the mouse showed a freezing response, and then as soon as they released from freezing and started walking around, there's a little blip in the activity of these cells, right? So it looks like you need these cells to turn on, potentially, for the mouse to escape. And that just shows all that summary data. Where the escape is initiated is the dotted line. You can see the activity is increasing a second or 2 before they initiate the escape. So then we can do a simple experiment and ask, what if we just silence these cells? What if we just functionally take them out of a circuit? What happens to the behavior? So we use chemogenetics, this approach that I talked about earlier, to silence these cells in vivo. And when we do that, this is a control experiment saying that these cells can express this reporter protein, YF, yellow fluorescent protein. There's no change in their behavior, so we get about 80% escape. And here's the chemogenetic experiment, where now you go from 80% down to about 37% escape when these cells are silenced, okay? So that tells us these cells are necessary for the escape behavior. We can do the same thing with optogenetics, so we have a little bit more temporal precision, so we can just switch these cells off. These are the cells with the control, about 80% escape. And then these are the cells, this is what happens to escape behavior when we silence these cells, right? More than half do not escape. So again, just a second approach to show us that these cells are necessary for this escape-like behavior. So it looks like what these cells are doing is they're responsible for creating a bias. And the bias is towards this active defensive escape behavior in these mice. When you take the cells out of the picture, the mice tend to show more of a passive response. Either they do nothing or they learn to freeze, okay? So we're going to use this model now to just explore a little bit more about the role of these cells and about how these mice learn. And that's the second part of this talk. It's called Learning on the Fly. And let me set up the talk in this way. So in your life, our life, the iguana's life, you encounter different types of risk and risk that has different value, okay? So if we think about risk of 1.0 as the risk that's like catastrophic and is going to end your life, that could potentially happen at any time, right? So most of your life is you have risk odds that are relatively low. There might be an event that's a little bit higher, but this risk could happen at any time. And so your brain is wired or engineered to always watch out for that catastrophic risk. You don't know when it's going to happen, but it could happen at any time. So what would be great is if you could learn from risk, right? If you were exposed to something really risky, but not catastrophic, so you survived, it would be great if you could use that to lower, right, that risk probability in the future, increase your chances of survival. Do these mechanisms exist? Yes. Where are they? How do they work? We don't know very much about this. So the idea is, can you use exposure to risk to kind of create this tingly spider sense where you know something's going to happen? So if we go back to this image that I showed you earlier, I showed that CRH activity increases before the run, continues to increase, and then goes down, right? So we think about this as a pre-flight phase and a flight phase. And so the question we wanted to ask is, what do these single cells look like in this pre-flight phase and this flight phase? This is these miniscope recordings. This is activity from single cells that are CRH cells. And you can see when you take all the trials that some cells turn on early, some cells turn on late, but there's this little cluster of cells that seems to turn on right around escape onset, right here. So we can look at single trials, and you can see right around escape, right before the execution, during the initiation phase and execution phase, there's like a brightening of some of the cells. Not all of the cells, actually very few of the cells. And you can see that the cells that turn on in the first escape are not necessarily the cells that turn on in the second escape, right? So there doesn't appear to be like an escape cell. These cells collectively figure out that they need to turn on, and then they drive escape. Skip this part. All right, so we silenced CRH neurons before, but now we wanted to say very specifically, what happens if we just silence after the mice have already started running? So you hear the footsteps, you're running. What if now we silence CRH neurons? Do you complete the run, or do you just stop dead in your tracks? My bet with Nuria was a coffee that they would complete the run, that once it had started, it would end. This is Catherine Simone. So Catherine was an electrical engineer who was doing a PhD in my lab, and she was amazing at like doing magic, which meant like creating all these cool tools that allowed us to analyze all of this behavior in a really kind of beautiful, complex fashion. And so using Catherine's tools, we could start to get a really good handle on the behavior, the tracking, where the mouse were, how they were moving. We started to pick up things like, this mouse would take off, but it would avoid the shadow. So we're starting to look at kind of path curvature and how actually they follow escape trajectories and things like that. So Catherine and Nuria built this really cool but very simple little device. In the arena, they created a situation where they had an IR beam that went across the arena. And as soon as mice started to run and broke this beam, it created a feedback loop that activated a switch that turned on yellow light that silenced these cells, right? So we weren't doing it, the mice were doing it to themselves. And so that's what this experiment is. You let them start running, then you turn off these cells, and you just watch and see what happens. So the first thing that happens is that if you track speed, you notice that as soon as you silence these cells, the speed drops off, right? So this is before the break, the speed is the same. This is after the break, right? So basically the speed is cut in half. But what are the mice doing? So it turns out when you silence the cells, that's this ARCH 3.0 data, most of the mice abort. So they start to run, we shut off these cells, these mice just give up. They stop whatever they're doing. They don't freeze, they just seem to drop their head and start walking around. So you can see escape rate went down, freeze rate, what didn't change, but it's the aborted rate that goes up, okay? So these mice have totally forgotten, I don't want to use forgotten because that implies something else, but these mice have totally changed their behavior just by silencing these cells. Using Catherine's tools, we could also start to optimize the path, as I've already shown you. So we could just follow the direction of the nose and see where it goes. 0 degrees is pointed straight at the shelter, and you can see that mice, when the light turns on quickly, or when the shadow comes, they quickly orient toward the shelter. And we can do this for a number of trials. Oops, sorry. And they quickly orient to shelter. But if we do this when we silence CRH neurons, which is at time 0, you can see their heading vector completely changes, right? They're heading to the shelter, you shut off these cells, and they totally lose the plot, right? So now these mice change where they're looking, they stop, they start wandering around. So it looks like these CRH neurons are required for path optimization. Before the break, they can go in multiple paths. After the break, you can see that normally they head towards 0, but if you silence the neurons, then this kind of path breaks down and starts to go in different directions. So it looks like CRH neurons are necessary for this completion of flight. If you allow CRH neurons to activate, the mice escape. If you silence them, they turn around and don't escape. The other thing we noticed is that these mice show this drift in where they sit before they escape. This is trial 1, and each of these plots where the mice are. The hotter the color, the more time the mice dwell in that area. Okay, so about 6 seconds they spend in this danger zone. In trial 2, they spend a little bit less time in this danger zone, a little bit more time in the shelter, a little bit less time. And by trial 5, they're not spending very much time. And the time they are spending is closer to here than it is to down here. Right? So rather than about 5.5 seconds, they're spending about 2.5 seconds now in this danger zone. If we silence CRH neurons, however, they don't do this. So they haven't been to the shelter, and now when they come back, they just kind of hang out in the same spot they were hanging out in before. So we think that this kind of drifting that's happening, right? This kind of drift towards the start line is not happening when CRH neurons are silent. So their time to break the beam normally from the onset of our trial drifts down to about 3.5 seconds. It remains at 7.5 seconds otherwise. You can also look at their nose positions at the different trials, which I showed you all of them. But you can see that in the naive mice, they slowly start to orient better towards the 0, right? But in these green mice, where we silence CRH neurons, they never, ever get this sense of direction or orientation, right? So these mice remain really, really poor at orienting towards their target, which is the shelter, and completing the task. So it looks like CRH neurons are necessary for action initiation, which I showed you originally in the first part of the talk, and responsible for action execution. So we think that they play this really important role in trial-by-trial learning. So the mice learn this on their own, we just watched. So we have two conditions in CRH neurons. When they're active, the mice start moving the start line for themselves. So they start to creep a little bit closer to increase their probability of success, which is reaching shelter. If we silence these cells, mice do not do that. They do not learn to cheat. They have the same potential probability for safety. Many of them don't know what safety is because they never completed a trial. All right, for the last kind of 10 minutes, I'm going to tell you about this new work that we've been doing. So the last work is not yet published. This work is not yet published, but it kind of takes us in a different direction. And all of these collectively speak about not the CRH neurons driving behavior, but creating a state for particular types of behavior. So we think a lot about threats that trigger behavioral and visceral responses. But most of the threats that we think about in the lab are these external threats, right? They're overt and explicit threats. We do foot shock, we have a potential predator, we expose them to the odor of a predator, we expose them to a mouse that is aggressive. So these are very explicit threats. But that's not how our world works, right? Most of the things that are potentially a threat or potentially a danger are like on this page, right? They are people. And I think our society has figured out that that's important, right? So you can go anywhere and somebody is taking a picture of you to figure out. And they're going to use the picture and your microbehaviors to figure out, are you doing something good or might you do something that somebody deems not very good, right? And so we have this kind of surveillance world already existing to try to detect potential threat. But it's something that we also are doing all the time, right? So we're always trying to figure out, where's the threat? Because we need to know which one's the bad guy, which one's the good guy, right? So how does the brain do this? And how do we solve this question? And especially, how do we solve it in mice that largely look like this? Very difficult, right, to understand just looking at mice, like, why would this mouse be a threat and not that mouse? But mice seem to know, and they know very quickly, and I'm going to show you some data that proves this. We're going to go back to this setup, the fiber photometry setup, and we're going to do something super, super simple. We're just going to put another mouse into a cage where there's a mouse. And here's what happens. If you put an intruder into the cage, the mouse we're recording from the mouse that's in the cage, it shows an increase in the activity of these CRH neurons, which is brief. It lasts about 10 seconds, and then it goes back to normal, okay? That's for all the mice, but I'm going to break this down a little bit more. So we introduced the mouse to an intruder it was familiar with. So its brother or sister. So we've done this in males and females, females and females. It was either a brother or sister, right? Kin. And when you do that, you see an increase in the activity, brief increase in the activity of these cells. Or we introduce them to same strain of mouse, but an unfamiliar mouse. So they'd never seen this mouse before. And now you see a little bigger increase in this activity. So we can quantify this, and you can see the unfamiliar activity is much higher. We can throw all the numbers up to show you the peak Z-score, the area under curve, which just integrates this information, right? To show you that if it's a stranger, these cells show a bigger response. Is it just novelty, right? Maybe it has nothing to do with the fact that it's another mouse. It's just different. So we did this experiment in which we exposed this mouse to either an object that it was familiar with or an object that was new. So you expose it to an object, come back with the same object the next day, or you expose it to a ball object and come back with a different object. And you ask, if it's just novelty, then the response to this brown box should be bigger than the response to the blue ball. And in fact, it turns out that's not the case, right? The response to the ball, the response to the box are identical, right? Suggesting that these neurons are not responding just to a novelty signal. They're responding to something else. Could they be responding to threat? Could they be trying to calibrate, is this mouse a danger to me? So we did that same familiar, unfamiliar experiment, and then we added a third arm, and that was to introduce an unfamiliar, but it was a much smaller juvenile unfamiliar. So the probability of threat is likely lower. And what happens when we do that is we find that the juvenile response is more like the familiar adult and much smaller than the unfamiliar adult. So we do think that these cells are calibrating some type of social threat. If we introduce a different strain of mouse, what happens? Is there a strain specificity to this response to unfamiliar? And the answer is no. So we introduced an unfamiliar CD1 mouse, which is usually aggressive. In our case, the mice did not fight. And so we just treat it as another strain, and it just responds to unfamiliar. So different strain mouse, unfamiliar equals unfamiliar, familiar equals familiar to these mice. Then we started to look at what they were actually doing, okay? So mice do all sorts of interesting little things. You just have to carefully annotate all of them, okay? So this just shows in a single mouse, over about a 3-minute period, all the different actions they engage in. And these are the actions that are engaged in by the resident mouse, the one to whom we present the other mouse. And you can see this mouse sniffs, it digs a little bit, might groom a little bit, not much. And here's when an unfamiliar mouse is presented. You can see the resident spends about half its time just sniffing the other mouse. And sniffing is one of the best ways for mice to get information about the other mouse. And we can do this in a bunch of mice, in a bunch of different trials, and unless you don't see the colors, there's a huge difference in just the baseline behavior of these mice. And all we did was introduce a mouse that was familiar or a mouse that was unfamiliar, right? So a massive increase in the time spent sniffing by the residents. We can break this behavior down into even smaller incremental parts. So mice show very canonical constrained behaviors when they socially interact. They show nose-nose interaction, they show a nose-tail, nose-torso interaction, they show a nose-anogenital interaction. That is because mice release signals in urine, right? And so these mice are detecting information from the urine. And then they show a chasing interaction where they just chase the mouse at the back of the tail. And so we can quantify all these, and each of these colors maps on to the different behavior. So pink is this nose tail, dark gray is anogenital, that light brown color is nose to nose. It always starts, almost every interaction starts with just a very momentary nose to nose contact, kind of a hi, and then the rest is a how are you. And this is all the data from every single mouse. These are just females. We've done the same number of males, but I just show you the females just because it's, otherwise the graphs get too messy. And you can see in the female familiar, they show all sorts of interactions, right? A little bit of anogenital investigation, some body torso, and it kind of ends by about the end of a minute, minute and a half in most mice. When it's an unfamiliar mouse, you can see the interactions are totally different, right? There's lots of dark gray, which is that anogenital investigation, which is to gain information from pheromones about the state of the mouse and who that mouse is. So we can add all this up and see which of the behaviors is actually different. And the only one that tracks as being different is the anogenital investigation. This is just a rolling 60-second average of investigation, and you can see that the purple, which is the unfamiliar mouse, is much bigger than the blue, right? We can also start to look at each of these individual events, and each of these is just a square every time there's an event. And you can see there's way more events when it's an unfamiliar. We can add them all up, and we can show that over the course of 150 seconds, there's way more of these anogenital investigations when the mouse is unfamiliar as when it's familiar. Actually, by about 20 seconds, the familiar mouse, they've pretty much become disengaged. But in this unfamiliar mouse, it takes over a minute to start to even plateau in this behavior and interaction, right? So they engage longer. It's a very specific behavior that they're trying to get information from. So our question was, well, what happens if we take these CRH neurons, right, these stress or emotion state control neurons, out of the picture using optogenetics? Well, the first thing that happens is that in the control, nothing happens, right? The unfamiliar, there's still more sniffing than the familiar. But when you silence these neurons, now there's no difference, right? They investigate unfamiliars and familiars the exact same. If we look at the specific behaviors, and specifically that anogenital investigation, we find that if it's a familiar mouse, silencing these cells does really nothing to that behavior, right? There wasn't a lot of this behavior to start with, about 10, 12 seconds. Now it's down to about 8 seconds. But in these mice, in the stranger mice, when it's an unfamiliar mouse that's being introduced, if you silence the CRH neurons of the resident, you completely drop this behavior, right? You go from about 30 seconds for each mouse to about 10 seconds, right? Looking more and more like familiar mice. So these neurons, we think, are really important in promoting the engagement of this type of behavior. They're not driving the behavior because, remember, they just turned on for like 10 seconds, right? But they're creating this permissive state for this behavior and for these mice to investigate. So through this talk, I showed you 3 different things showing you that CRH neurons seem to be really important in biasing towards a go signal rather than a no signal in response to a predatory-like threat. CRH neurons appear to be critical for allowing these mice to exhibit this really interesting form of trial-by-trial learning, which is very subtle. And they also seem to guide how these mice approach and interact with other mice, specifically when the other mouse is a stranger. So I'll go back to where I started from in the talk, which is thinking about this idea of whether stress is an emotion. And so emotions, if you just Google emotions, you get a definition that's something like this. Physical and mental states brought on by neurophysiological, so brain changes, right? Variously associated with thoughts, feelings, behavioral responses, and a degree of pleasure or displeasure. That's pretty like generic vanilla definition of emotion. There's no scientific consensus on the definition of an emotion, which is kind of interesting, and I think speaks to how complex it is. Emotions, as we know, are not limited to humans, right? Darwin described this over 100 years ago in the expression of emotions in man and animals. If you have a pet, you know exactly what I'm talking about. Your pets can tell you about their emotional state very, very quickly, and you will learn about your pet's emotional state very, very quickly, as they will learn about your emotional state very, very quickly. And that the neural correlates of emotion still remain unresolved. So David Anderson and Ralph Adolphs about 10 years ago put out this paper, which was kind of a call to arms to start to think about, are there specific criteria that we need to lay down that help us identify the neural correlates of an internal emotional state? So they said 3 things are really important, that they integrate signals from different sensory modalities, including innate and conditioned stimuli. So I just showed you sensory modality visual, which is the looming shadow, the mouse, which is visual, but then also an olfactory stimulus. They said it was necessary and sufficient to drive a diverse set of environmentally appropriate defensive responses. I showed the necessity or the necessities side. We did not show the sufficiency side. They said that activity should be required to instruct the memory of an experience, which we think they do in this trial-by-trial learning. And finally, they said that the fulfillment of these criteria implies that a structure is not just a relay station of a sensory input, nor does it just drive a specific motor response, but occupies this kind of middle ground, this intermediate position, right, that gives rise to a state, this feeling, this something that has human emotional value and likely changes how you engage with the world and the behaviors that you exhibit. And I think stress seems to serve a lot of these functions, and specifically these CRH PVN neurons, we think, have a lot of properties that are consistent with a key cell cluster that confers emotion state-like properties. So this is the figure I started with, and this is the figure that I'll finish with. If you think about stress as an emotional state, but not just an emotional state like a happy or sad, but an emotional state that has lingering effects well after the stress is gone, I think that type of framework helps us think about a lot of these things very differently, rather than when you just think about stress as an explicit response to a challenge that triggers a cascade of physiological and behavioral effects. So I'll stop with that. Thank you very much, and happy to take questions. Yeah. Oh, actually, we'll let the students go. Yeah, go ahead.
Me?
Yeah.
Hi, amazing talk. Thank you very much. I'm just curious whether there's a reason you're calling it stress and not fear. Is there a difference between the two in this case?
This is a very long semantic discussion around fear. Yeah, I think even fear, a lot of rodent biologists don't want to even refer to fear as a thing. The reason I refer to this as stress is because we can lean on the fact that glucocorticoids or corticosteroids go up in response to just about any stressor. And we always see a responsive activation of CRH neurons and an increase in corticosteroids. So that's why, and we've kind of come from stress world, so we continue to refer to it as stress. The encoding of fear is not happening in these cells. We've looked at very explicit fear behavior. So example, when people induce a fear-like response to a tone and a shock, right, a freezing response, that response has nothing to do with the cells that I'm looking at. So I think that allows us at a cellular or circuit level to try to tease those things apart. The core dynamics of fear and the storage of fear are distinct from this. Yes.
I also want to say that was a beautiful talk.
Thank you.
Thank you for being such a good communicator. My question is about those animals that didn't run away. You had quite a large portion, 15%, that actually did nothing.
Yeah.
So what's special about those animals' brains? What do we know about their wiring? Like why are these animals not doing anything?
Yeah, this is a great question. So we have noticed this, that the non-escapers seem to cluster, right? It's not like this trial is not an escape and this trial is actually, we have some mice that are resistant or reticent to escape. We don't know, and it's very difficult for us to compare neural activity across mice because we have to normalize to the baseline in that mouse, so we can't take the absolute numbers. I think maybe with single cell data, we might be able to tell if there are more cells or fewer cells that are being activated. But my guess is it's not going to be down to the level of these neurons. I think there's a few things. One could be, it could be something really simple. These mice don't see as well. I don't know. You know what I mean? Like there could be a lot of very early things where the signal is just not even relayed. And I don't have a handle on that. But it is something that's in the back of our mind, and we have enough trials now. I think we've probably done like 1,500 mice across my lab and Nerea's lab, that we could start to think about, go back to the videos where they don't escape, and just ask, like, are they just inattentive? Do they look up at the shadow? Do they make a particular type of movement? Yeah. So I think that might be, because then that might allow you to do a different set of experiments in that cohort of mice, right? The non-escapers. Yeah.
Thank you.
Thanks. We'll go here, here, here. Yeah.
I was wondering if you
Yeah, this is a really good question, and we're going to do this. We're going to do these experiments. We haven't done this. This is brand new, but we do want to expose the resident to other things and then ask, how does it respond? I can tell you one very quick thing. If the resident is stressed, has just been stressed, it is not interested in the intruder. It is dealing with whatever it's dealing with, and it's not interested in whoever's walking in that door. It does not investigate at all. Yeah, but yeah, this is a really, really, really good question. Open, yeah. Yeah.
Are you planning on
Yeah, we've thought about this experiment a lot. I don't think we have the right tool. So here's the problem with optogenetics and the way that anybody does it. So when you get these cells to express channel rhodopsin, for example, or any of the optogenetic constructs, it's very difficult to control how many cells express it, right? So that's a variable. And the second is that when you turn them on, you synchronize their activity. And you could see from the miniscope data that it's not synchronized activity. It's like some cells turn on, then other cells turn on, then other cells turn on. And we don't really have a good way to do that. We could, so we have been thinking about using something called a step function opsin, which is an opsin that when you turn on light, just depolarizes the cells a little bit to reach more of an active state, so makes it more permissive to fire, doesn't drive the firing, and the cells just do this whatever way they want. So I think the prediction would be that in the step function opsin, that you would have higher escape probability in those. We're already at 80%, so we don't have a lot to play with. But the step function opsin may also kind of increase stranger danger, right? Or it may make familiars appear to be strangers. So those are, but we need to be able to temporally control that. And with the step function opsin, you can turn it on and you can turn it off. But we just haven't gone down that road because I think it'd be like a year of just testing the tool, and I don't want to do that just yet. So yeah, but it's a great question. Thanks. Yes.
Hi, Dr. Bains. Thank you so much. In a group, and I'm curious, like in that case, would you anticipate individual for both mice, or would it be
Yeah, that's a really good question. We've never, ever done that. We haven't thought about that. And the reason I'm thinking about this is just, I think about our arena and I think about the logistics of that experiment. So I think there's a way to do that experiment. I think you need at least 3 escape routes. So I think you need 3 shelters. You need more shelters than there are mice. I think if there's just one, I'm not sure what would happen. Right? The one closest may go and the others may, I think, would freeze. That's my guess. We've never done the experiment. I would like to move to a more naturalistic arena, something that's like more like this. But it's a trade-off, right? If the distance is too long, they're not going to run. They're going to make the calculation. I'm just better off hiding in the corner and hoping that nothing happens. Yeah, but that's really interesting. Yeah, I'd never thought about it. Yeah, it's a challenge. The analysis is also a challenge to track 3 simultaneously when they're all the same color and not lose the identity, the digital identity of the mouse is non-trivial. So yeah. It's an interesting thought though about whether you can have some type of social learning based on what others do. Yeah. I think though we would have to have a consequence. So right now in our model, there's no consequence. They escape or they don't escape, nothing happens. Right? So in that model, it would be interesting to see if the ones who don't escape, there's a consequence for them, right? They get grabbed by some, something that is like a bird or a hand or whatever. And the next time they might learn to escape. I don't know. But yeah, good thought. Yeah.
In the stranger danger experiment, I was wondering if you
Yeah, the intruder doesn't care about the resident. So this is really, really interesting. Whoever is like in the place first controls the interaction. It's fascinating. I could take a mouse, I could put it in a new environment for 5 minutes. Whoever comes in next, this mouse is still the boss of the environment. It's like if somebody walks into your house. Somebody walks into your house, I bet you say hi first. They don't. There's 0 chance. Unless it's like, surprise, like they might do that. But for the most part, and it's wild with mice. You would think that in some environment that neither mouse has been in, it shouldn't matter if this mouse got here a minute before this mouse. But it always matters. Always, always. This mouse always does the questioning. That's what we think this interrogation is. We even call it interrogation. It's questioning. Who are you? What are you all about? Are you a danger to me? Right? That's what's going on. Yeah, but yeah, we've looked, and in a paper in 2018, we looked at the other mouse. It's a very sparse behavioral repertoire. Like they do a little bit, and they're like, whatever. So, yeah. Yes.
Let's do one last.
Last question at the back.
I was wondering if the responses of these
Yeah, good question. So we do 5 trials. One time, that's it. We have done a second set of trials in a mouse just to exactly ask this question. Not with we didn't have the photometry. We were just looking at the behavior. And if we do a second set of trials a week later, we get only about 50% escape. So it's kind of a chance now. So it's not that they freeze more, it's just that they don't do anything. And I think it goes back to the earlier question, there's no consequence. And so they figure out, whatever, like it's a cloud, right? It's not a predator for them anymore. So yeah, thank you.
Thank Dr. Bains for the fantastic talk.
Thank you.